So we've been through several parts of the scientific method that include observations, hypotheses, and experimental design. A very important part of experimental design are variables. Experimental variables are factors that produce the cause and effect that you're studying and that you suggest or predict in your hypothesis. So selecting these variables is a real art form when you're designing your experiment. There are three types of variables: controlled variables, independent variables, and dependent variables. Let's go over them with some examples. Controlled variables, and any variable is a factor that can impact cause and effect. Controlled variables are factors that do not change during the course of the experiment. For example, if you're testing COVID methods for detecting COVID antigens, you might control the variable for the cotton swab technique. If you are looking at medications that control blood sugar, in your experiment, you might control the amount of sugar in the liquid and the volume of the liquid that is drunk before a diabetes test. And if you are testing fruit production in a field, you might control the variable for the soil composition in that field. So it might be all clay in that field or all silt in that field. Independent variables are the second type of variable. Independent variables are factors that you do change as the researcher. So for that COVID antigen test, you would have an independent variable of changing into new antigen test methods that you want to evaluate. That would be an independent variable. On your diabetes test, you might want to evaluate new medications to reduce blood sugar. So you would change between one medication and the other new medication. Or on the field, you may change the types of fertilizers that you use between one row and another row. Dependent variables are the third type of variable. Dependent variables are the factors that change due to the fact that you change the independent variables. So, in your COVID antigen test, the dependent variable is the number of true COVID positive test results that you get. It'll differ between your test methods that you're evaluating. You hope it differs. In your diabetes test, your blood sugar levels will change dependent upon the medication that you use for your test subjects. For your agricultural test, the fruit production in a field will change based upon the different fertilizers that you used. So three types of variables. In a well-designed test, only one independent variable changes at a time. So for instance, in your antigen test methods, you may want to change the test method and the gender of the people that you're testing. But if you change both of those variables at the same time, then you won't know if the cause and effect is due to the gender change or the test method change. So you change one independent variable at a time in a well-designed experimental study. In addition, experimental designs have test subjects that are divided into groups. And those groups are either experimental groups or they are control groups. Experimental groups get the different independent changing variables, and they have data recorded as a result of those. Control groups do not get the changed variable. Control groups are groups where we know, based on previous work, we know what to expect. And it helps us to clarify the fact that the change in the independent variable probably produced the change in the dependent variable. Let's explain that a bit more. So when it comes to the control groups, we are using variables that we know what to expect from the data. In some cases, we know not to expect a change in data, and in some cases, we know to expect a change of data. The best way to show you this is to really talk up a sample, an example. So let's talk it up. If you are collecting data for positive COVID results or blood sugar levels or fruit production masses, and you have independent variables of new test methods or new medications or new fertilizers, then in your control groups, you will not use those independent variables. Instead, you will use things that you know will produce a change. For instance, you know by using a very well-studied and accurate COVID testing method, like the antigen test, the PCR test, you know that you'll get a certain amount of COVID positive results because it's very accurate and it's tried and true. So by subjecting the control group to this method, the PCR method, you know you're going to get a change in your data to reflect the positive COVID test results. This is called a positive control group because you're expecting a change to note the positive COVID results. Let me give you another example. If you are looking for blood sugar levels and you're looking for a reduction in blood sugar levels with new medications, then the best way to make sure that your new methods are producing validated results is to compare them to a method that you know very well reduces blood sugar by 20%. Because if you can replicate the data that shows with your very well-known medication that you're reducing blood sugar levels by 20%, and your new medications might reduce blood sugar levels by 12% and 23%, then the combination of all that data is validated by this known response that you have here. It's a positive control group because you are producing well-known blood sugar results. Another example. With the fruit production, if you want to really understand how your new fertilizers impact fruit production, you'll compare it to a positive control group with a known fertilizer that boost production by 20% or 25% or 40%. Because that way, when you get those numbers hit right on with your data, then you can feel better about the data numbers that you have for the new fertilizers. These are positive control groups where you expect a change, you see the change, and it helps validate your other data. There's also negative control groups, and those are groups where you expect no change. So when you do see a change, you can relate it to your independent variables in the data. Let's go over that a little bit. It's actually pretty easy. For instance, if you were looking for COVID positive results, well, if you use samples with no virus, then you wouldn't expect any COVID positive results. That would be a negative control group. You'd have no change in the number of COVID positives that you have. The classic negative control group is placebo medication. If you're looking for a reduction in blood sugar and you use a placebo, you will notice no change in blood sugar. It's a negative control group. And if you're looking at fruit production in a field, your negative control group is the group or the row that uses no fertilizer at all, because you expect no change in the amount of production with no fertilizer. So these are control groups versus experimental groups and different types of control groups. I hope that helps you out, because control groups are very important to saying that the data change that you noticed is directly impacted by the independent variable change. This delta is for change. So it really helps to validate your result and your experimental design. Remember, just because data correlates with a change doesn't mean that that change caused the data change. You know, correlation does not impact causation. You've heard that phrase before. Correlation doesn't necessarily mean that what you changed caused the change. But when you design an experiment well with controlled groups, you come much closer to demonstrating that a correlation could imply a causation. See on the next slide.